Alright guys, so today I'm going to try to explain a little bit more about DNA structure and the process of replication and hopefully make the complementary base pairing process a little bit clearer for you. So I've got a little flip chart we're going to look at together here, and we'll start with just basically um, why is DNA so important? So hopefully we remember from previous lessons and stuff that our chromosomes are made of DNA and our chromosomes are found in the nucleus of the cell. All right, so chromosomes, remember they look kind of like little X's or whatever, and they are basically really tightly wound DNA. So chromosomes are made of DNA. And then DNA, what we're learning this week during this unit is that DNA is a double helix. So we see those two strands of nucleotides there. And then the DNA is made up of sections called genes. Okay, so each of these sections is a gene, and the genes code for specific proteins. Okay, so that's kind of how this all is going to work together, because once we get finished talking about DNA, then we're going to bring in RNA, and RNA helps in this process of actually taking the DNA information and then converting it into RNA, and then eventually making proteins. And then the proteins control all the things that happen inside our cells and control the traits that we have and things like that. All right, so that's just kind of an overview. So when I ask you, what is the pathway to a protein? It always starts with DNA. Okay, so DNA is the first step, and then next the DNA has to be converted to RNA, which you're going to learn about in one of your PowerPoints, and then that eventually the RNA gets read and proteins are made. Okay, so that's the pathway from DNA to proteins. Okay, so and that'll make more sense to you after you watch the PowerPoint with the RNA. All right, so let's go ahead and oh goodness, let's see. Here we go. All right, so right here, what I've got is showing you what a nucleotide looks like, and I'm hoping this is just a review, because we should know what a nucleotide looks like. Remember, it's made up of 3 parts. It's got the 5-carbon sugar. In this case, the sugar is deoxyribose. Okay, I'm just going to write deoxy for short, but deoxyribose. And then we've got the phosphate group, and then the nitrogen base. Now, the nitrogenous or nitrogen base, there are 4 of them. Okay, and so this part right here is the part that can change or it can vary. So if I ask you what part of the nucleotide changes, well, it's the nitrogen base, because it can be adenine, or it could be thymine, or it could be cytosine, or it could be guanine. Okay, so there are 4 different nitrogen bases. Now, within a DNA molecule, the sugar doesn't change and the phosphate group doesn't change. The only thing that changes on the nucleotides would be the nitrogen bases. All right, so this diagram right here, what this is showing us is how this all comes together. So see each of the nucleotides. See, here's a nucleotide, and then here's a nucleotide, here's a nucleotide. Okay, and over here we got the same thing. So these are nucleotides. Oops, okay. So notice G is pairing with C, T is pairing with A, C with G, G with C, T with A. Okay, so that's our base pairing rule. Okay, so if you don't know that, if you were struggling with that, if you looked at that daily grade and you were like, what in the world is this? Then you need to write this down, and this is what you need to make sure that you memorize, and that you it's like one of your main things you got to know for this unit. Okay, so the base pairing rule is A is going to pair with T. T is always going to pair with A. And this is when you're talking about DNA to DNA. See, like this is a DNA strand and this is a DNA strand. So when it's DNA to DNA, A pairs with T, T pairs with A, C is going to pair with G, and G is going to pair with C. Okay, so write that down somewhere so that you have it, and that'll help you when you're doing your daily grade and everything. Okay, so that being said, let's go ahead and look at the next slide. Okay, and this was in our notes. You had one example problem in your notes. So I want to do another one with you just to make sure we've got this. Okay, so look, let's read the problem together and let's highlight a little bit of key information here. So this is Chargaff's rule, and that's what we just went over. A with T, T with A, C with G, and G with C. That's Chargaff's rule. That's complementary base pairing. All right, so you're going to have 400 microgram sample of DNA. So that's your total amount right there. Okay, and then you've got 60 micrograms of cytosine. So I've told you how much of one particular nitrogen base you have, and from that you can figure out everything else. So what you're going to do is you're going to write down the 4 letters. So you've got C, G, T, A. Those are the 4 bases. Well, in the problem, I told you there's 60 micrograms of cytosine, so you'll write 60 for that one. Well, we just learned that Chargaff's rule is C is going to pair with G. So if C pairs with G, that means they are always going to be in equal amounts in the DNA sample. So that means you automatically know that you have 60 micrograms of guanine. So now you're going to take those two and add them together. So 60 plus 60 is 120. Okay, so now you're going to take that number and you are going to subtract it from your total amount. So you would say 400, which is your total, and you subtract the amount for these 2 bases, which is 120. So 400 minus 120, that gives us 280. Okay, so let's stop and think about that. 280, is that our answer? Is that just for adenine? No, that's the amount for thymine and adenine. So what we need to do to this number is we've got to split it between these two. So we're going to divide 280 by 2. So 280 divided by 2, that gives us 140. So that means you have 140 micrograms of thymine and you have 140 micrograms of adenine. So the answer to the problem is, because it's asking for adenine, is 140 micrograms. Okay, so just remember, you always take your smallest number, add it to itself. That's what we did there. Okay, and then take that sum, subtract it from your total, and then whatever you get when you subtract, you divide that by 2, and that's going to give you the answer. Okay, so that's how you do those problems in case you were struggling on that at all. You'll have at least one of those on the test, so you do need to know that. Okay, so now the last thing I want to go over is DNA replication. And DNA replication is just when DNA makes a copy of itself. Okay, if you replicate something, you copy it. And hopefully you remember that DNA is kept in the nucleus of the cell. So if I ask you, where does DNA replication occur, you would tell me the nucleus. Now, if I would take this piece of paper and I wanted to make a copy of it, if it's all rolled up, am I going to be able to copy this on the copier and get a very good copy of it? No. Okay, so what I would need to do is unroll it, right? Unwind it. Okay, so that's the same thing with DNA. DNA, okay, I'm going to make this window just a little bit bigger because I want to show you something. I normally, when I teach this in my class, I usually act it out, sort of. So, DNA is 2 strands, okay? That's my arms. It's twisted up. It's a double helix, okay? So, it's 2 strands of nucleotides. I'll twist it up into a double helix. Well, it cannot be copied this way. It has to unwind. Then, and it becomes like a straight ladder, then the strands are going to separate from each other. And then when they're separated, then you're going to have free nucleotides that come in and they're going to pair up with the exposed nucleotides on each strand so that you end up with 2 molecules of DNA. Each one has an old and a new strand, and they twist back up. Okay, so let's kind of draw a little picture of that. I'm going to try to use some different colors and stuff to help us. All right, so our original DNA molecule, it looks something kind of like this maybe, all twisted up like a double helix, okay? So it cannot be copied that way, so we have to, it's going to unwind, okay? Now these lines are just representations, okay? So they're representing those nucleotides with the phosphate groups and the sugar and the nitrogen bases. I'm going to write some letters here. So we got A, T, T, G, C. Okay, so over here we got T, A, because remember our complementary base pairing rules, right? A, C, and G. Okay, now in between those nitrogen bases are hydrogen bonds. So that's what I'm going to draw right there. So there are hydrogen bonds. So if I ask you what kind of bond holds the nucleotides together in a DNA molecule, it's hydrogen, okay? All right, now those bonds have to be broken. So an enzyme called helicase comes in and it's going to split break those bonds. And so now the two strands can separate from each other. So that's what's going to happen next, is the strands are going to separate. And some of this stuff is happening simultaneously. So, I mean, the way I'm drawing it, it seems like it's step, 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 step. But actually, as they separate, the next process is kind of happening with it. But anyway, all right, so we got it. Let me rewrite my letters here. We got A, T, T, G, C. And then over here, we got T, A, C, and G. Okay, so when they separate like this, now they're exposed. Now all this is happening in the nucleus, and there are 3 nucleotides that are just waiting for this process to occur, and they're going to come in and they're going to pair up with their complementary nitrogen base. So A is going to pair with T, T is going to pair with A. Hopefully you're doing this along with me and you're like, oh yeah, I know that. T is going to pair with A, G is going to pair with C, and C is going to pair with G. And then there's one molecule, okay? And it would have hydrogen bonds between those bases. Okay, then over here, T is going to pair with A is going to pair with T, A is going to pair with T, C with G, and G with C. And there's our other molecule. Okay, and it's got its hydrogen bonds that are formed. So now we have 2 new molecules. Here's one DNA molecule. Here's the other DNA molecule. And notice each one has an old strand. The red would be an old strand. And then the blue represents new. So each one has an old and a new strand. Then these would, the last step would be that they would twist back into a helix, which is kind of hard to draw. Okay, so something like that. Okay, so we call this, because each molecule has an old and a new strand, we call this a semi-conservative process. All right, so think about it. Conserve. If you conserve something, you save it. Okay, so during this process, part of the old DNA molecule is being saved. Okay, so each molecule has an old strand and a new strand. And that's why we call it semi-conservative. It doesn't save all of the old, right? It's not all old. This molecule is not completely old or completely new. It's semi-conservative, so it's one old strand and one new strand for each molecule. Okay, so that's DNA replication. It occurs in the nucleus, and hopefully you remember it occurs during the S phase of the interphase, okay, of the cell cycle, and it is a semi-conservative process, and the whole thing is all about making copies of the DNA. Okay? All right, now the last thing I want to show you real quick is kind of what you did in the little daily grade. And if you were struggling with it, then hopefully this will help you. So let's say that your DNA strand is T, A, C, G, G, T, 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 A, G. Okay, so all you would need to do is do the complement. So remember our rule. T pairs with A, so that's what we go here. And then A pairs with T, and C is going to pair with G. And then G, G, T, well, that's going to be C, C, A. And then T, T, A would be A, A, T. And A, A, G would pair with T, T, C. Okay? So when you're talking about DNA replication, this is going to be different. We're going to learn something else with the next PowerPoint. So you're going to see a slight change. But when you're talking about DNA replication, this is the rule. T pairs with A with T, C with G, G with C. Okay, so I hope that cleared some things up for you. If you have any questions, feel free to message me on Schoology, and I'll try to reply best that I can. And I've been doing some video conferences. I haven't had too many of you take me up on the offer, but I'm here if you need me, and I'm posting these videos, so I hope you're watching. Okay, y'all take care, and I will see you soon.